recordings. There we go. So we can let folks who can't be here catch on YouTube later. But thank you everyone for joining. We're here with, I'm Natasha Barlota, the Stewardship and Outreach Manager from the National Loon Center. And then we're joined by Jacob Job from the Bird Genoscape Project. And I'm going to switch you, Jacob, over to the host so that you can start sharing your screen.
Okay, and then I'm going to, Kristen is there. So my co-host, everybody, is Kristen Basu from the Genoscape Project, and she is trying to get in here, and I am trying to let her turn her screen on. Natasha, do you know, should I make her a host or?
Yeah, if you make her a host, you should be able to get her screen on there too. Then we can spotlight hers.
There we go.
Well.
Kristen, you should be able to, there she is. So I'll do an introduction for myself, and then I'll let Kristen introduce herself, and then I'll get into the talk. First of all, thank you to the National Loon Center for inviting us to give this webinar about loon genoscapes and everything we have to talk about. Like Natasha said, my name is Jacob Job. I am the Associate Director of the Bird Genoscape Project, and we are located on the campus of Colorado State University in Northern Colorado, in Fort Collins, Colorado. Kristen, do you want to jump in and just say hi and introduce yourself and then turn it back over to me?
Yeah, so hello everyone. My name is Kristen Basu. I'm the research scientist with the Bird Genoscape Project. I've been working with Kristen Ruick for about 5 years now. So I do a lot of the data analysis behind many of the genoscapes and a lot of the offshoots of the projects that we're doing. It's great to be here. I'm excited to talk about loons.
Thanks, Kristen. And Kristen really is the muscle or the machinery behind what I'm going to talk about today. So don't let her be too humble. I'm just the talking head here. She's the genius behind a lot of it. So keep that in mind. So I'm going to go ahead and share my screen so that you all can. Well, I can't share my screen now. Let's see.
I think we have to switch you back over to the host and then.
Oh yeah, switch me back. That would be good. And there we go. Should be able to share it. There we go. So everybody can, someone confirm you can see my screen?
Yep.
Okay, great.
Yep.
All right. So really, the goal today, I mean, we're going to talk about loons, which is goal number one. But really, we want to talk about the goals behind the Bird Genoscape Project, what the Bird Genoscape Project is, what a genoscape is. So we're going to give you a crash course 101 into understanding that. And then once you have a pretty good understanding of what a genoscape is, what we do within the Bird Genoscape Project, we'll direct our attention to applying that to Common Loons, looking at what we've discovered about the loons, their populations, and the threats that they face, and which populations face the most pressure from those threats. And then we'll wrap up with a little bit of application, and then open it up for questions and comments and whatever you all might have to say. So using genetics to understand threats to common loons. So this is a genetics talk. Again, I don't have a background in genetics. I have a background in acoustics and conservation biology. So if we get into any of the technical questions, we certainly can answer those, and Kristen can jump in and help me out. I think the best way to sort of talk about the Bird Genoscape Project, at least the way that most people might be familiar with or understand, is through the lens of ancestry.com, or really any sort of genealogy type service that have become so popular over the last decade. Ancestry.com is just one of those services. But the idea is you can take some of your DNA from spit swab, hair, blood, whatever it might be, and you send it into a service. They analyze that DNA, and from that, they're able to know beyond the fact that you're a human, but what your roots are. Where does your DNA come from around the world? And you can see on this particular map, which is not an all-inclusive map, but they show you in these different colored circular type structures the different genetic populations, so to speak, around the world where your DNA may have lineages to. And so with the Bird Genoscape Project, it's ideally or essentially the same thing, except we're working with birds instead of humans. If we kind of look at an example of what that might look like with a bird, we'll take you in a little Bird Genoscape Project 101. We have a yellow warbler here. And what we know about yellow warblers, or what many of you might know about yellow warblers, and what the field guides say about yellow warblers is this is their range on the right side of the screen. Can someone confirm that you can see my mouse moving? Just shake your head up and down. Yeah, you can see it. Okay, so this is the range of the yellow warbler. Anything here in orange is the breeding range of yellow warblers. Anything in yellow is part of the migratory range, migratory route, and anything in blue is the wintering range. That gives us a understanding of the species as a whole. But if you really want to work on protecting yellow warblers, for instance, in this case, we have a problem in conservation, in the conservation world, where we have a limited amount of energy, we have a limited amount of dollars. And so we really want to take those limited resources and apply them as directly as possible and efficiently as possible to have the greatest impact. And so basically what I'm saying is we need to find out a way to sort of dissect this overall yellow warbler range into unique or separate genetically distinct populations. So the question is a yellow warbler a yellow warbler? So to do that, and this is how we do our work with all species, we'll use, again, yellow warbler as the example, but the unit of measure where our work begins is a singular plucked feather from a bird. So if you're at a banding station and we have bird banders across the Western Hemisphere, Canada, United States, Mexico, Central America, down into South America, they are tasked with capturing birds. They apply their bird banding tags to them. They do whatever else they do with those birds, but we've asked them to do one other thing for us, and that is to pluck a tail feather, generally the outer two tail feathers of those birds. The reason we ask that is, just like if you were to pluck your hair and send it off to a genealogy service, at the base of that hair is some of your DNA in those skin cells. Well, at the base of this feather down here, and this picture just kind of bothers me, I'm realizing now, because you shouldn't be holding the feather from this end, because that's where the DNA is from this yellow warbler, and you could be contaminating it with these human fingers, this human DNA. But at this side of the feather, the calamus, the part that sticks into the bird, this is where the DNA of the bird is. And so once we have that feather, hopefully we have that DNA, enough of the DNA to do our work. So we gather enough feathers from across the breeding range, in this case of yellow warblers. The other way we can gather DNA from birds is through blood samples. So if we're not collecting feather samples, if for some reason, you know, the banders are working on another project where they're collecting blood, or they prefer to collect blood. And honestly, blood is easier to work with for a few reasons that I won't get into. But this is another way we can get a hold of the DNA that we need to do our work. And so you can see they've punctured the brachial vein of this particular bird. The blood is going into that capillary tube, and then they'll put a cotton swab over top that puncture wound, close up that wing, and within a minute that puncture wound will be sealed up, and the bird will be good to go on its way. And you can actually see on this particular bird, they've already applied that unique band on its leg, so it has a unique number associated with that individual. So these birds get caught. We know where they get caught. The people that do the catching, they send us the coordinates, the latitude and longitude. Then they send us that DNA, and then those feathers make it to Colorado State University, where we are housed. And so what we're showing you here in this particular picture are some of our undergraduate workers. The feathers come to us in these coin envelopes. The coin envelopes have a lot of metadata associated with them. They have, what species feather is in there, the location where the feather came from, the sex of the bird, the age of the bird, all sorts of information that we need to know to be able to do the work that we need to do. And so this particular shipment, you can see that is being worked on. It's quite large. There's probably several hundred feather samples from all different species that came to us. And then the students go through and they take all that metadata and they put it into a form that we work with. It gets uploaded into our database. And then once they're all sorted through, those feathers get put into these boxes and they get stored in our freezers for long-term storage. Just give you an idea of what we're working with here. So this is our director, Dr. Kristen Ruig here, front and center. This is Christine Rain in the background. She is another person who really does a lot of the behind the scenes of making the Bird Genoscape project work. So I wanted to highlight both of them because they're instrumental, obviously, in this work. But what you also see in this clearly posed photo are a lot of these coin envelopes with bird feathers inside of them. And then these photos here in the front and center, you can see the feathers on top of the coin envelopes. And I was just thinking before I jumped on this call, so these coin envelopes are 4 inches wide. We now have over 200,000 feathers in our collection. So I just did a little back of the envelope math, and I'm pretty sure if you were to lay all of our coin envelopes back to back next to each other, we have 12.5 miles of feather samples in our collection, which I don't know if that's impressive or not. It's just a fun little number I wanted to throw at you guys. And then here's another picture of what the feathers look like when they are stored in our collection. So we have American robin samples here on the picture on the left. 06 means these feathers probably came to us in 2006. And they sit in these freezers until they're ready to be used to build a genoscape or some other purpose. On the picture on the right, or in the picture on the right, this is where we store our DNA or our extracted. So the DNA that gets extracted from blood or tissue, the DNA gets put in tubes and they get stored in our -80 freezers. And this is a look inside of a freezer that's at -80 degrees. And yeah, just like it sounds, it's really cold. You can see it sublimating that frost coming right off of it. So that's just a little insight into what it looks like to sort of handle or store all of these feathers. Once we've decided that we want to build a genoscape, and by genoscape, I think I mentioned this, but I'll mention it again, is a sort of a breakdown of the breeding range of a species. So what are the distinct genetic populations just in the breeding range? And so once we've decided we want to build a genoscape, we will go into our collections, we will pull out feathers from across that range of that species, feathers that are representative of that range. And then the work begins to get the DNA off of those feathers. So we cut the tip off of that calamus. The DNA is stored there. Christine really is the one that does the magic of, I will call them potions because I don't know what the chemicals are. But there is a process, a very precise process that she goes through to remove the DNA out of those skin cells, to isolate the DNA. And then those samples then get shipped off for sequencing, so that we can understand what the makeup of that DNA is. And so this is just like a look into some of our lab space to kind of bring to life what the lab space at the Bird Genoscape Project looks like. So let's jump back just for a brief moment. I won't go into this too much. This looks a lot more scary than it really is. But if we go back to high school or maybe even middle school biology, remember that our DNA is essentially just a string of 4 letters, A, T, C, and G. The combination of those A's, T's, and C's and G's across our entire genome, the human genome, the way those A's, T's, and C's A's, T's, C's, and G's are put together is what makes us us. They're what makes humans humans. And that's the same thing for any species on Earth. It's just the combination of those 4 letters and how many of those 4 letters that exist. So what we're trying to do, or what we do with the Bird Genoscape Project to sort of isolate those breeding populations into genetically distinct breeding populations, we're looking for differences in the sequences of A's, T's, and C's and G's that tend to group together in places around the hemisphere. So if we go back to our ancestry.com example at the beginning, if you send off your DNA to the genealogy lab and it comes back and it says, okay, you have roots in Northwest Europe, you have roots in Eastern Europe, and you have roots in North Africa, it's because there are certain differences in your DNA that are represented in those regions. So people with also from those regions have those differences as well in their DNA, and they lump together. So that's essentially what we're doing. Those differences lump together in breeding populations of birds. So let's go back to our yellow warbler. So after we've done all that work, we've collected feathers, we've extracted DNA, and we've run the sequencing. What we have found with yellow warbler is we have a genoscape here. So this entire map with all its colors is the genoscape. Again, this is the breeding range of yellow warblers. And we have found that there are 5 genetically distinct populations of yellow warblers, meaning each of these 5 different colors, they have differences in their DNA that have lumped together to say, okay, I'm a yellow warbler from the Southwest population, or I'm a yellow warbler from the purple Pacific Northwest population, or I'm a yellow warbler from the East, so on and so forth. So these are what we sort of call conservation units. If we go back to what I said, remember, we have a limited amount of time and money and effort in conservation. If we want to apply those limited resources most efficiently, we can now say, I'm making this example up, but bear with me. Let's say the Southwest population of the yellow warbler is not doing very well, but the rest of the populations are doing quite well. They're expanding their populations. Well, before the Genoscape, we would have had a limited understanding of where we would need to apply those limited funds, those limited resources. But now that we know we can separate these populations from each other, we can say, okay, we need to go to these places to do the work, to apply those resources to help protect or conserve yellow warblers. And so really that's the idea behind this work. If we take it a step further, so once we've constructed the Genoscape, we can ask our partners across the Western Hemisphere, especially in Central and South America, to start plucking feathers from yellow warblers, any yellow warblers that they gather. They can send those feathers back to us. We can sequence them. And we can now say, okay, these birds that were caught in the wintering range, where did they come from? So if we look at, I'm trying to move this, there we go. If we look at these pink samples, so I think this is Colombia, these pink samples that were plucked from yellow warblers in Colombia, they were sequenced and it came back and said, well, the birds that winter here, they came from the eastern population up in United States and Canada. Anything in light blue on the wintering grounds, those feathers were plucked, we sequenced them, and it came back that they spend the summer, they breed in the central part of the US. And so now all of a sudden our partners down in Central and South America, they know where their birds come from. So if we go back to the made up example that I just gave you, let's say our Southwest population, which is doing poorly in this example, their populations are declining, we can now apply those limited resources in an even more fine-tuned way. So maybe those populations are doing just fine on the breeding grounds, and it's really on the wintering grounds where we're seeing those populations start to shrink. Well, now we know where to go look and apply those resources on the wintering grounds, anywhere there's a red dot on this map. And so we're really fine-tuning our approach and learning a lot about these populations and how we can apply conservation resources more pointedly. Let me take a quick drink real quick. And Kristen, I should say, if you feel you need to jump in and clarify anything, feel free, because I really like a conversational style type presentation. All right, so enough about yellow warblers. None of you are here because you want to know about yellow warblers. Maybe you like yellow warblers, but you hear about the loons, and I'm here about the loons. And might I say, just like real quickly, I'm from Michigan. I spent my summers vacationing in northern Michigan growing up. I heard my first loons, I think when I was 12 years old, fishing on, I think it was a long lake in Alpena with my dad. And so they're my favorite species on earth, bird or otherwise. I have the passion that you guys have. It's crazy how much common loon stuff there is in my house. So this is special to me to do this presentation with you guys. So that's really why I was excited to do this. Enough about me, back to the loons. So, okay, we've walked you through an example. And so you're familiar with the range map of common loons here. Again, anything in orange, the breeding range of a common loon. Anything in yellow is a migratory range or migratory pathway of common loons. And then anything in blue is where common loons spend the winter. But can we do better? Can we tease apart those genetic populations? And the answer is, well, hold on. Let me introduce our partners first. Sorry. So we had, and you all may be familiar with Alec Lindsay on the left, Michael Sorensen on the right from Northern Michigan and Boston universities, respectively. And the Biodiversity Research Institute, they reached out to us asking us to address that question. Can we tease apart those populations of common loons? What are the conservation units of common loons? And so they had worked for a long time. Alec had collected samples from the 70s of common loons. And then Michael and him were working on the genetics, but couldn't quite, figure it out. And we had actually this process figured out by this time. And so we were able to step in and sort of help them answer these questions. And the Biodiversity Research Institute was interested in understanding the impacts to common loons across the range. And so it's kind of a perfect storm of all this coming together. And really, this is a model we use for any of our genoscapes. Individuals or groups have questions. We have the tools genetically, we can help answer those questions. And so we form a coalition to focus on that goal. And that's what happened here with common loons. And so to give you just a few numbers, I don't want to get into the nuts and bolts of that. Sample collection occurred, like I said, 1979. The samples go as far back. Most of the samples were collected from 1992 to 2016. To construct the Genoscape, remember the Genoscape, we're just talking about breeding birds. 560 samples were collected. In the case of common loons, we're not talking about feathers, we're talking about blood samples. So 560 individual loons were caught and blood was taken from them. When we're talking about migratory and wintering loons, 252 samples were collected. And it was a combination of blood, which I just mentioned, which was collected from living, healthy birds. Blood or feathers were collected from dead or moribund birds. And then we collected tissue samples from carcasses on these migratory or wintering grounds, carcasses, birds that were determined to have died either through botulism poisoning or they were impacted solar sites. And so we had three types of samples: blood, tissue, and feathers. Still, the DNA was there that allowed us to do this work. And really that highlights sort of the power of our approach is we don't always have to have living birds to do our work. We can try to salvage DNA from dead birds to uncover questions about impacts, what caused their death, where they came from, and what populations are being impacted. So the big reveal, why everybody is here. What does common loon population structure look like? And here it is. So you can see from this picture, we have 6 genetically distinct populations, breeding populations of common loons. Again, these conservation units. 6 different colors: green, pink, red, yellow, blue, and purple. And if you squint closely, I had to draw a circle around this one. If you're familiar with common loons, there are isolated breeding populations in Wyoming, Montana, and Idaho. And genetically speaking, they share ancestry with this central Canada, this red population up here in central Canada. So I just wanted to point that out because it's kind of hard to hear. So I won't get into whether or not this is a remnant population or anything like that, because I don't feel like I can speak to that. Maybe Kristen can at the end if anybody has any questions, but just wanted to make sure that that was visible. Okay, that's question number one. How do we divide that breeding range up into genetically distinct breeding populations? Question number two, where are these populations wintering and what routes do they take to get to their wintering grounds? And so first, there's a lot going on here. So let's just focus right now what's in the black circles. And right here, we have all the birds that we got DNA samples from on their migratory routes. Anything that was in yellow, these little diamond shapes over here, they came from this breeding population around the Great Lakes. Birds in this maroon color, it was determined that they had come from the central Canada breeding population. And then if you look down here in the desert Southwest, again, we see the central Canada population largely represented with these red maroon triangles, but we also see some of the Great Lakes birds migrating all the way across the country to the Southwest. And so we had a few birds that were sampled over here, which I thought was interesting because that's certainly not the shortest route as the crow flies. Our wintering populations that were sampled, okay, same thing we just talked about. So over here on the West Coast, we have birds largely from the central Canada population. We have a couple of birds from that Great Lakes population. And then we have a couple of birds from this sort of Pacific Northwest population in pink right here. If we go over to the Atlantic coast, over by off the coast of Massachusetts, Maine, New Jersey, we see most of those birds are coming from either the Great Lakes, the yellow population, or from this Northeast population in purple. And then if we go down to the Gulf Coast, where so many loons spend the winter, we have samples only from the the yellow Great Lakes population or the maroon central Canada population. Notice that we don't have any of the population from Alaska, the green population, anywhere on this map. And it's not because they aren't wintering in any of these locations. It's likely because we just didn't sample any birds from those locations. And it could be that these other populations, the blue population, again, you don't see it anywhere. It's just likely that we didn't collect samples from those populations. So that's why you're not seeing them represented on the wintering grounds. But one of the things you should have been asking yourself at this point is, why are these shapes different? Or why are these shapes different? Circles, we've got diamonds, we've got triangles. What does this all mean? And those are getting at the threats that exist to common loons that we looked at in this particular study and where those threats exist. So there's a lot going on here. You can pretty much ignore everything on the right, this legend right now. I left it there just to refer back to. But if we just focus on botulism right now. So in this study, we collected samples from a number of birds that were determined to have died from botulism poisoning. Those are those birds that are in this black circle in the center of the map. And so, yes, we know common loons were dying from botulism here in the Great Lakes region on their migratory routes. But we can take it a step further with this Genoscape approach, and now we know the relative impact to those, which populations were impacted most. And you can see that most of these birds were birds that were from the Great Lakes anyway, that they were breeding in the Great Lakes regions. But some of them coming from the central Canada region, making their way across the Great Lakes, which stopped over to rest, refuel, were impacted by these botulism outbreaks and also died. And so we can see that we can tease apart or fine tune what we know about the impacts of botulism on common loons by having this Genoscape. If we turn our impact to solar energy, so these birds were also birds that died on their migratory route. So if you're wondering, what's going on here. Birds aren't wintering down here in the desert Southwest. They just hadn't made it to their likely wintering grounds out here on the Pacific coast or Baja California. Instead, they encountered and were impacted by solar sites all across the desert Southwest. So they either flew into a panel, landed on a panel, something happened, and interacted with wiring, something that killed these birds. And so we can see that these Great Lakes birds that were breeding over here, they decided to migrate all the way across the country, and unfortunately, they collided with a solar site, and 3 of them died here. But many, many more from the central Canada population were impacted by these solar sites and died. And so we can start to tease apart what are the relative impacts of solar development on the different populations of common loons. And then finally, we have a bunch of birds that we don't know how they died. We don't know what happened to them. But what's useful about this is we can go back and try to uncover the reasoning for these deaths. Once we have determined the cause of deaths in these birds, we can then apply the same knowledge that we did with botulism with solar sites and say these birds were from the purple region of our genoscape. This many were impacted by this cause of death. This many were impacted by this cause of death from the Great Lakes region. And then the same thing down in our wintering populations around the coast of Florida and in the Bahamas. We can do the same thing. And so just because we don't have the answers right now doesn't mean we can't find them in the future and then understand which populations are being most impacted. So that's a lot. I tried to distill it down into the points that I really want you to take away, which is essentially common loons do have these genetically distinct breeding populations. A common loon is not just a common loon, no matter where you see it. And then we also can tease apart sort of our understanding of which populations are being impacted most by what causes, so either disease or human infrastructure, whatever it might be. And so this work is in this publication, if you're interested in reading it, that you see on the screen right now. Those are all, not all of them, but a lot of the individuals that were involved. Well, this is all the individuals that were involved in the authorship of this particular publication. There's certainly not everybody that was involved in the process of getting those samples and then coming up with this paper. And then these are the main groups, the main players in that process that you see down here. And so really, I put this up here because it really and truly is a team effort. None of this work is done just by us. It's impossible to do this work just by us. And so I really want to give credit to all of those different team members because they really do play an integral role, each and every one of them. And that, and I mentioned this earlier, that is the model that we use in every one of our genoscapes. So I just want to spend a little bit, I just have a little bit of time left let's focus in on the renewable energy question. Remember those solar sites that we talked about, the loons that were impacted by them. This is something that we're really interested in doing for all species, not just common loons. There's a lot of potential here. If you think about a bird that impacts a solar site, typically these sites are in the desert. They go through a lot. We don't always find their carcasses right away. And a lot of times they can be scavenged on by different animals. They get desiccated and dried out and they fade in the sun. And we're not always able to determine what species was impacted. Sometimes a bird may hit a solar site. It may not die, but it may leave behind feathers or what we call feather spots. That's still information that we can use to understand what are the impacts of solar sites on migratory birds. And so we've come up with sort of this workflow that represents what we are doing. And so that's, you don't have to memorize all these details, but the idea here is we have a solar site, birds are being impacted by these solar sites, and they're leaving behind either entire carcasses or just those feather spots that I mentioned. The feather spots are particularly of interest because oftentimes we have no idea what species they are. Well, we're able to with our technology now to determine those species with the DNA on those feathers. We can look at that mitochondrial DNA. And then once we do that, we can then match up that DNA with the particular breeding population, the genetically distinct breeding population in the genoscape. And once we've done that, so we can take those feathers, we can place them on the map, we can then look at sort of relative impact. In this particular example, the orange population is impacted more than the blue population is impacted more than the green population. And so for any species that we have a genoscape for, we can start to better understand how are solar sites impacting them. And then with that information, we can then turn it over to the managers of those sites and say, you know, when these species are moving across the area, when they are potentially in danger of impacting these sites, maybe we can take measures to lessen those impacts, whatever that may be, you know, positioning solar panels a different way, or turning mirrors in a way so they're not shining at the tower at Ivanpah. I don't know if you guys are familiar with it. I can't remember the name of this type of solar, but you've got hundreds of mirrors pointing at this tower, which is warming a substrate, which is generating power eventually. But what happens is birds can fly through these lights and get singed, or they will die. And so what are the steps that these sites can take to minimize these impacts when they're most needed? Or before these sites are even set up, we can understand, okay, what species are moving through, what populations are moving through this potential solar site, and is it a good idea to put a solar site here? If not a lot of birds are moving through this particular site that are of conservation concern, then maybe it is okay to put a solar site here. But if a lot of birds of conservation concern are moving through this particular area, maybe it's not the best idea. So these are the sort of decision-making trees that we're helping to inform with this approach. And just to show you some examples, what that might look like. So this is common yellowthroat. These colors, the red is a genetically distinct breeding population in California. The green is another genetically distinct breeding population. And each of these circles represents a solar site that is currently in existence in California. And it shows you the breakdown of which populations the common yellowthroats that were impacted at these solar sites are coming from. So Ivanpah, there was one common yellowthroat that was killed. Here at Mojave, there were 6. McCoy, there were 3. Desert Sunlight, 4, and so on and so forth. You get the idea. Almost all of the common yellowthroats that were impacted at these sites come from this west population, the green population. But it looks like one of the birds from the McCoy site came from the southwest population. So that's how you read this map. We did the same thing for yellow warbler and Wilson's warbler. You could see, especially for Wilson's warbler, the variation impacts at these different sites. Eritricity, we have 3 different genetically distinct breeding populations that were impacted, or Wilson's warblers that were impacted. Same thing for McCoy. There are actually 4 different breeding populations represented here. And so you start to understand the relative impacts. But again, none of us are here because of common yellowthroats, yellow warblers, or Wilson's warblers. We're here for common loons, and we've done some of that work out in California. I've showed you that in a prior image, but now you can see the breakdown of those impacts. So at the Genesis site, there were 4 common loons that were impacted. 2 of them came from the Central Canada population. 2 of them came from the Midwest population. Desert Sunlight, 1 of the birds out of the 8 came from the Midwest population. 7 of them came from the Central Canada population. So just a wealth of information that we've never had before that we now have access to. And you can imagine scaling this up across all solar sites across the country, really anywhere in the world. We could do wind sites. I mean, the possibilities are really limitless, and we're just laying forth a pathway that we think is productive in helping with conservation. And then the last thing I'll leave you with, which is good because I'm starting to run low on time. We have partnered with National Audubon Society to help them produce their Bird Migration Explorer app, which is now online as of, I think, mid-September 2022. You all should go there if you haven't been there. It's a wonderful website with a wealth of information that has 2 goals. It's allow people to connect their migratory. My dog is growling at something outside. If you guys hear that, that's not my stomach. It allows people to connect the migratory birds in their backyards to other places around the world. So if a common loon was seen in a lake in your backyard, let's say in Missouri, we now hold that information potentially to know like where that bird potentially could have come from, all the way up to maybe the Alaska population. A bunch of different groups. You can see all the different contributing members down here have used different methods. We use genetics. There are radio telemetry tags. There are bird banding labs. I'm missing so many obvious things right now, the other tools that are out there, but the idea is it's a central repository to put together this migratory information. The second goal, though, is to give you an idea of the threats that all of these species face along their migratory routes. So the Alaska population of common loons, if you were to do a search for that species in the Bird Migration Explorer, or just common loons in general, you would see along their migratory routes down to their wintering grounds, what sort of threats do they face along the way, and what impact is it having on common loons? And so they've deemed these conservation challenges. So this is something that's just come out. Not a lot of people know about it yet. We are one of the contributing members of this with the information, our genetics information that we have developed. So I encourage you to take some time to investigate that. With that, I will just say thank you for your time. Again, this is quite an honor just because I can't emphasize enough how much I love loons. It's almost to a weird level how much I love loons. But I hope you all do too. I think you all do, because that's why you're here listening to this talk. And with that, I will let either Kristen jump in or open up the floor to questions. Oh, and real quickly, if you want to know more about the Bird Genoscape Project or get involved as a contributor, you can visit our website. All of that information is there, including my email address if you want to reach out to me and ask me more specific questions that you can't find on the website. Oh, if you want to ask questions, put them in the chat. It looks like that's how we're doing this. And Kristen, I'll let you know. Actually, Kristen, before I get started with those questions, is there anything you wanted to add, or are you okay at this point? Oh, she's muted. Natasha, can you help me with that maybe, or do I have to do that? The Bird Migration Explorer is the app. I don't know how to bring back Zoom fully. Bear with us just for a second. We'll get to your questions. Oh, I see. Okay, I got it. You guys are probably like, who is this guy? There you go, Kristen. I think you've been unmuted.
Great. I can just jump in if there are any questions that you want to pass over to me. But
okay.
Yeah.
Okay. So how do loons get botulism? You know, so botulism essentially develops from a growth of algae on these bodies of water. And it's just, I don't know if it's a natural byproduct of algae, dying algae, live algae, or what, but essentially the warmer these bodies of water get, the more algae is present there. And the more algae that is present, these are, I'm pretty sure, blue-green algae, the more of them that are present, the more the botulism, the bacteria associated with botulism are present. And so when the loons stop over on these lakes, they're drinking water, they're eating food. Basically, the bacteria is getting inside of them, and they're getting sick from it. And I'm pretty sure it causes neurological issues, but I could be wrong on that. Do wind turbines affect loons during migration? You know, I don't have the answer to that. I don't know that we have the answer to that. It's plausible, certainly, because these are objects on the landscape that birds could collide. Birds can and do collide with. Whether or not loons are doing it, I don't think we have that answer. But I wouldn't be surprised if they are impacted by.
Can I jump in on that one?
Yeah, go ahead.
Yeah, so a lot of our focus is on different solar sites, but we do have hundreds and hundreds of samples from Altamont, which is a wind turbine. I want to say one of the largest in California. And we have not seen any loons there. Like, had we had that, that would have been a really nice, we would have actually sampled and we would have genotyped those birds. So we are predominantly seeing loons at solar sites, which is not unheard of given they're water birds. So like one of the thought processes, like you're seeing more water birds at these solar sites because of the lake effect or like the shimmering effect of the solar panels. Like if they're flying somewhere, they need water, they might be confused about what they're actually landing on. They might think it's a body of water where it's actually a solar field. But we have not seen loons impacted by wind turbines as of yet.
Yeah, that's I think it's called the lake effect hypothesis with these solar sites. Essentially, they emit, or they can reflect UV light back up. So if you're a bird flying over the landscape, UV light often is an indicator of a body of water down below you, because bodies of water emit UV light back off of them. And so you can imagine a bird could be confused thinking that an array of solar panels might be a body of water, especially when they're flying at night. So mistakenly think they can land. And then if you know about the biology of loons, they can't really take off from land. They need bodies of water as sort of runways to get started and to take off so they can get stranded. The bird migration, the migratory app that I mentioned, I think I said this, but it's the Bird Migration Explorer from Audubon. It's a wonderful tool. There's a lot of information there. You could spend a lot of time exploring that, but I encourage you all to do so. With your background in auditory with loons, do you think there's a way we could use sound to disorient a loon so we could help capture it when needing rescue? So I will say that one of the tools that scientists use to often capture birds is doing playbacks of their calls. And that's especially effective with male birds who might mistakenly think that a speaker is another male on its territory. It'll fly in to investigate, and it can be caught in a mist net. Other ways it could be captured. With loons, I'm trying to understand when a loon might need rescuing. Is it on land or water? I'm not quite sure what that means. But certainly, I think we have the capability of using the sounds of loons to lure other loons in. And I'm almost certain I have listened to a podcast where there is a group of loon biologists doing just that at night, playing the sounds. The male or the breeding pair comes in to investigate. And then they were able to capture those loons and perform whatever work on them that they were trying to do. So that is a possibility, and it's something that certainly happens. Any other questions? Okay. For loons, the secondary feather and breast feathers are typically taken for mercury and other sampling during necropsies or finds of dead loons not collected in the field. Are those sufficient and appropriate to test genetically? Yeah, that's a great question. So the reason I mentioned at the beginning that we typically pull tail feathers off, we're doing this largely with songbirds, so birds that are small in size, and the tail feathers, first of all, they're quite large, which means they hold more DNA on them than the body feathers. But we've also found that the tail feathers, when we remove them, the ones that we remove, has little to no impact on their ability to go about their business after we let them go. With the loons, almost assuredly, since it's a large-bodied bird, we would be asking for body feathers rather than tail feathers. So I don't know if it would be a secondary feather, which is a wing feather. That's actually probably larger than what we would ask for. I would imagine we would ask for breast feathers, just because of the size of the bird. So I don't know, Kristen, if you know what feathers specifically are taken from water birds, but it seems likely it's body feathers, not tail feathers.
Yeah, I think I'd be more curious. So a lot of times we share feathers with people doing stable isotopes. So they get a portion of the feather, we get the DNA part of the feather, and splitting it up. I'm not sure what you actually need to do the mercury testing for what you're talking about. But as Jacob mentioned, we're working with very small birds, so our priority is to get as much DNA as possible. With the loons, we would be okay with breast feathers. It would work. We would be able to get DNA from that pretty easily.
Yeah, and the key is contamination. I mean, there's environmental DNA everywhere. These loons are in the water. There's lots of different species DNA is likely on them. But when we have collaborators take feathers, we ask them to do so in a way that doesn't touch the end of that feather that pokes into the bird. And we have sampling protocol on our website if people are interested and contributing that you can go download and better understand how that process looks. So when they're hooked in fishing, so this is Scott. I think this is you're referring to your previous question. Oh wait, maybe not. So oh, Linda, yeah, this is your previous question. When a loon is stranded, when they're hooked in fishing gear and are diving, saying something not a loon call, but something else to maybe distract them to be able to net them. I don't know. I'm not a loon biologist. I suspect it is possible to, and I suspect scientists do distract birds in other ways than just playing their calls, but I can't speak to that directly. Natasha, I don't know if this is a question for you. I will let you answer this from Scott. It says, how can the National Loon Center work with these results now and in the future to possibly aid loon populations? I will say before, if Natasha wants to jump in, I will say that all of our information is publicly available. And so if the National Loon Center wanted to reach out to us to get this information and to do more work with the DNA that we have, they're certainly able to do that. And we are willing to work with any group that is trying to use our information that we put out there for conservation purposes.
Yeah, that is a great question. This is where we're starting at, I think, with trying to use these results. When Jacob reached out to me and shared about the project, I hadn't known about this, and it was something that was really fascinating. And so I thought we have to try to get this out to more people, because it's really cool. I learned so much about loons, knowing that there are some loons from the Midwest that are going down to the down to the coast of California there. That's not what we previously had known, as far as I know. So as a center that's focused on research and conservation education, we want to get this type of information out to the public. And so starting with this webinar and other educational materials to get out to people, including in the center that's going to open, that's a place where we want to be able to feature cutting edge research. And I've talked to Jacob previously about, is this project ongoing? Can we contribute samples? Because we would also like to know more about the loons here in Minnesota, and in the Midwest, more about them on their wintering ground. So a lot of potential for the future.
Next question. If one were to find a deceased loon on your lake, who should they contact? I think the best role, and we don't have this sort of workflow worked out with the National Loon Center, but I suspect they have the knowledge for what to do with that better. We're really not interested in collecting whole dead loons, carcasses in this particular instance. But we certainly could be interested in the feather samples from some of those loons, but we would have to work out that process with the National Loon Center to make sure the appropriate permits are in place to be able to collect the loon, the specimen, and also transport those feathers across the country to our labs. This all takes a lot of permitting. That is a pain in the butt, but highly appropriate, and is really necessary to do this work. So I would answer that. Natasha should probably lead the way on that one.
Yeah, I was gonna say, if this person that was asking is in Minnesota, the National Loon Center, we are facilitating necropsy research, and so you can reach out to us, and we facilitate the transport down to the University of Minnesota Veterinary Diagnostic Laboratory for necropsy. If the person is elsewhere in the country, it'll depend on where you are. Out in the Northeast, they have various loon organizations that are doing this type of work that you could be connected to, and that we can help you be connected to it as well. But yeah, it would really depend on what state we're talking about.
And I think we're done with questions, but one other thing I wanted to add is on the Bird Genoscapes Project's website, birdgenoscape.org, there is a page for the Common Loon. So if you go there, you can see these maps, you can see who our collaborators were. You can download the paper for free and read that. You can read a summary, and you can also, I think, gain access, or at least see where more samples came from, and possibly gain access to the genetic data. And so we try to make that information available. So if you really want to take a deeper dive and spend more time with this information, that's all there for you to access.
Jacob, I'm gonna throw in one more question for you on the one where you did mention you were going to go too in depth, and I'm going to ask a question regarding the Yellowstone population, the loons out in that area that are they're smaller and isolated, and it seemed like they were genetically from that
Central Canada.
Yeah, but geographically the Pacific Northwest is closer, right? So I guess that was one of the things that when I first read the paper and was talking to you, I thought this is really interesting to see and think, why are these loons down here? How did they end up there, and how are they probably more genetically related to a population that's a bit farther away than their closest neighbor?
Yeah, I mean, Kristen, this may be good for you, but I suspect there have been so I would start with, I'm sure we don't know the answer to this. I suspect glaciation played a role in this, is one of the hypotheses. So as the glaciers formed and maybe populations got separated from each other, I don't have a great answer for that. Kristen, do you?
I don't. I would almost wish Alec Lindsay was here, because he was also, like you, super excited about this finding, and he had an understanding of loons and what that interpretation meant. And I was the one who might have created the map, but I didn't have the loon biology underlying the patterns that we saw. So
Yeah,
reach out to Alec Lindsay. He's a good guy.
yeah, and actually, Alec he has a research lab in Northern Michigan, and reaching out to him, he might have some better ideas. But I suspect we don't, as a scientific community, we probably don't really know. So yeah, that's partly why I didn't want to get into it too much, because I don't have a lot of answers. But like you said, you pointed out the interesting thing: this population geographically is closer to this Pacific Northwest population than it is to this Central Canada population, and yeah, that's pretty interesting.
Oh, I do want to throw out that, at least from what I understand, and maybe it's still much closer to the Pacific Northwest, but that used to be a contiguous region. So the historical Common Loon range did extend further south, and it's currently further north, and so that is a remnant population. So it is probably something historical that happened, so they had this movement and maybe differentiation. But I still feel like even in the contiguous, even in the southern portion, if the Pacific Northwest existed, it should be a remnant of that, but it's just not. It's more closely related to the Central Canada population.
I hope that's somewhat satisfying, but it's probably completely frustrating.
That's just the beauty of science, is that there's always more things to investigate, always
Absolutely.
more questions that pop up.
Yeah, absolutely. I don't think there are any more questions. So I will just sign off, and then I'll let you close it out, Natasha, by saying again, thank you for inviting us here. It's been a pleasure working with you and putting this together. I really hope everybody has enjoyed this, has learned something about Common Loons, and about, honestly, migratory birds in general, what we are trying to do. So the goal of the Genoscape Project is to create genoscapes for 100 of the most threatened migratory birds across North America, and so that's what we are working on, and we're marching towards that goal pretty quickly. And so stay in touch with us to see what other birds genoscapes come online in the near future. We've got a bunch that we're working on. Yeah,
thank you so much for talking with us and sharing about the research. This was super interesting. I learned a lot more in this webinar. Well, in addition to reading the paper and talking to you before, there was even more that I learned here today. So it was cool to hear you talk about it. And it's great timing as we're thinking about the common loons coming back up now to think about where they're coming from and from the wintering grounds, where are they coming from back up here? What's going on their migratory pathway, and how are these different populations of genetically different populations moving around North America? So it's just really interesting, and yeah, definitely makes me wish I paid more attention in genetics.
Well, and for those of you who are listers, who have life lists or backyard lists or whatever, now you have a new way of thinking about check marks. You've got common loon on your list, but do you have all 6 genetically distinct populations of common loons on your list? And
Yeah.
so it's another fun thing to do if you're not really interested in the science of it, but you want to add some more to your list. This is a fun thing people can do.
It definitely isn't. I don't know. Birders love lists and life lists and checklists. So I have to start a new loon list for this. Yeah. Thank you so much again, and thank you for everyone for hopping on. I do plan to put this recording up on our YouTube page, and also put out a blog about this as well, and do some social media outreach. So look out for those, and please share with folks that you think would be interested.
All right. Well, it's nice being with you all. Have a good night. And Natasha, we'll be in touch.
We definitely will be. Everyone, take care.
Bye.